I'd like to start with an acknowledgement that this initiative bubbled up from a thought process that was pulled together by Mike Boehm when he was president of APS, and it has emerged into this topic that we're calling Phytobiomes. So it's a process that came through the membership and now is being honed into a process of policy advocacy. So what is the phytobiome? Well, if my SWAT teams from the Public Policy Board have been doing their job, you all know what the phytobiome is. The phytobiome is the entire microbial community in, on, and around the plants. So tops of plants, bottoms of plants, roots. And this community is not only inside the plants, the endophytic compartments, but also outside the plants, the exterior of the plants, the rhizosphere. It is the community that influences the plants or that is influenced by the plants. So it can lead to better nutritional qualities of the plants, but also healthy plants or more food, feed, and fiber. It includes bacteria, archaea, fungi, oomycetes, nematodes, and other living organisms that surround the roots and influence the plants. So you can see that now we're looking at a more holistic, system-wide scheme. The initiative, the Phytobiomes initiative, is meant to target an understanding of how that associated community influences the plant or is influenced by the plant, and how we ultimately can use that information to improve crop productivity, quality, and safety. So the goal of this initiative is by 2025 to have a comprehensive understanding of phytobiomes so that we can accelerate crop production and improvement. So where are we starting? And I like to think about this as the old story of the blind man and the elephant, where historically we've looked at individual compartments or individual, like the rhizosphere, or we've looked at the leaves of the plants, and we've interpreted the plant and the process, or the phytobiome as we now are calling it, as either what organisms are associated with the surface of the leaf or those that cause disease. But really, it's not just the individual organisms. They act as communities and they act as groups, and they influence the plant in that way. So historically, while we've only been able to look at one or a few organisms at a time, now we need to start thinking this as a system. What about the communities of the organisms, and how do they influence not only the plant, but other microorganisms in that community? This, of course, includes cultured and non-cultured microbes, because we've not been able to look at those uncultured microorganisms before, and yet we knew and we know now that they do influence the health of the plant, the health of the soil, and the health of the environment. We're also interested in how the whole system works. Before we've looked at how a microbe causes a disease, or a microbe benefits a plant by acting as a biocontrol agent. But those organisms function together, the plant and the microbes, differently under different environmental stresses. So what happens? How does drought factor into this? How do high temperatures factor into this story? And these are questions that historically we've not been able to address holistically because we haven't had the tools to do so. So can we integrate this whole system to understand it? Why are we looking at the phytobiomes now? And that's because the technologies, we've had the emergence of new metagenomic enabling capabilities, which allow us now to look at this whole system, to think about it in a much bigger picture. We have high-throughput sequencing technologies, computational biology, the genomics, and other omics technologies. What these are allowing us to do is to assess the community composition, its function, and its activity. So we're getting a much bigger picture. The story is becoming much more complex, but that complexity is what we live with as we try to improve crops and how those plants produce. So how do we do this, and how are these technologies enabling us? So you can initially start with a soil sample all the way up to the fruit or the shoots or the roots. You can extract the DNA, so this is one level of complexity that you can look at. And you can amplify that by PCR, amplify a component of the genome, and that will allow you to determine who's there, what's in that community, the species level, across diverse organisms, fungi so far and microbes, but hopefully in the future, nematodes, whatever else is in that community. Or you can do whole genome sequencing in that using a metagenomic approach and get at what do the genomes of the organisms in those communities encode. What are they capable of? What's their genetic potential for improving the soils or the plants or or inhibiting other microbes, or what's their potential to cause problems? The other approach is to look at what they're doing in that community, and that's looking at what genes are expressed, and then either the protein or the metabolites that are produced by those genes. And these technologies would involve the metagenome transcriptome sequencing, the metaproteome analysis, and the metabolome analysis. So if we look at it from a simplistic point of view, we can ask now who's there, what can they do, and what are they doing in those communities. Very powerful advances. So I'm going to just give you a few examples of phytobiome studies that have already been published and are perfect examples of how we can use this information and how we can move forward. So this is a study by Ottensen et al., where they looked using a sequencing of PCR fragments at who is there, who's out there in that community. And they did a very detailed analysis of different plant parts. And by just looking at the different colors of the pie charts, you can see that the microbial communities, and this is bacterial communities in this case, are very different on the top of a leaf, from the bottom of a leaf, from the fruit of the plant to the root of the plant. Very different communities that are impacted and that are impacting that plant and are influenced by that plant. They also looked at where those microbes are distributed on the plants, because this can tell us what impact that community might have on that different set of organs. So, for example, if, in the color scheme here, you can see that the Berknea are very abundant on the lower parts of the plant, but not so abundant. I'm sorry, I have it the other way around. Very abundant on the upper parts of the plant, but not so abundant on the lower parts of the plant. Very powerful bits of information when we start to think about how these organisms are influencing the plants. What's really wonderful is we're not limited to prokaryotic organisms anymore. We can now ask these questions with eukaryotic organisms, and this is an example from that same paper where they looked at the fungal communities and where they were distributed on the plants. So again, very different communities on even different upper parts of the plants, let alone in the soil or root regions. In a couple of studies that came out a few years back, they tried to understand the core rhizosphere microbiome and how that core microbiome influences or is influenced by the plant. So what they did is they collected the DNA from the microbes in the soil, in the rhizosphere right around the roots, and then inside from the endophytic compartments of the root. And they asked, what are those populations that are in those different environments? So what did they find? They found that many of the microbes that are abundant in the soil were not found in the endophytic compartment, suggesting there's some selection going on there, as we might expect. And that the endophytic communities from soils were different, from very different parts of the world, were surprisingly similar. So the plant is selecting for communities that are similar from very different root environments. And that the host genotype, in fact, had a role in selection of these microbes. So the bottom line is that these communities are not just products of random assembly, but that these communities are maybe even predicted. So we might even be able, by the way, to breed for better communities. So can we breed plants, for example, that select for a beneficial microbiome? Or can we breed plants that restrict the not so beneficial microbiome? And have we inadvertently, through our breeding processes to date, because we've been selecting for plants on very highly fertilized or highly watered or high tillage soils, have we inadvertently selected away from an ideal microbiome? Lots of interesting questions that I'm going to pose. I, of course, don't have the answers for any of these questions, and that's what the initiative is about, to drive research toward these questions. So you've already seen our wonderful disease triangle once today, and I think all of you are familiar with this. But this is a very simplistic look at what we do as a discipline and the interaction with other disciplines. We need to have a conducive environment to get disease, and we need to have a virulent pathogen. But now what we're learning is that that is just too simplistic, that I like to say we're really talking about genome-genome interactions. And if you think about a cell or an organism is the product of its genome, and how it interacts with other organisms in the environment or in the context of its own tissues is an interaction of genomes. And those genomes influence what the responses are. And all of that is related to what the environment says it can do. So if we now turn to looking at plant microbe interactions in that context, looking at what is the influence of disease on the phytobiome, the structure of those communities around a plant. This is a study by Trivedi et al., where they extracted DNA from bacteria in the rhizosphere, and then they did two types of sequencing. They did the amplicon sequencing, which enables them to ask who is there, what genus and species are in that community. And they also analyzed the functional genes via a hybridization technique. And that enabled them to say, well, what can those organisms do? It's not saying what are they doing, it's asking what is the genetic potential of that population. And they did this on trees with citrus greening versus healthy orange trees. And so if we come back to my context of genome interactions, they're asking, does the genome of the pathogen affect the genome response of the plant, which then affects the genome content or function of the microbial communities surrounding the rhizosphere? So what did they find? Really very interesting piece of work. They found that the rhizosphere communities on the infected trees were quite different from those communities associated with the healthy trees. That, in other words, disease is associated with detectable shifts in the phytobiome. They also found that there was reduced functional diversity in the rhizosphere communities. So what that says is that in the rhizosphere communities that are associated with the infected trees, I didn't finish my statement there, I guess, with the infected trees, there was less composition of genes that had different biochemical functions. And the relevance of those biochemical functions is that there was a shift away from the use of easily degraded carbons by these microbes in this community to sources that are more recalcitrant. This is consistent with carbohydrate partitioning that's related to photosynthesis. So photosynthetates going to the roots is dropping. So in other words, they are able to, using a phytobiome approach or looking at the microbiome that's associated with this rhizosphere, provide more precise insights into the mechanisms and the processes of disease and the consequences. And by extension, using a phytobiome approach, more able to understand what's going on during resistance, predicting the mechanisms. Furthermore, from an approach that's useful for information in terms of diagnostics of the course of disease, you can identify microbial indicators of disease in the microbiome, or alternatively resistance. So looking at the more complex level is a very nice study by Baker et al., where they looked at the complexity of not only plant communities, so mixed plant communities. They then looked at individual plants and their influence on the rhizosphere. And then they looked at, so that would be plant-plant interactions versus plant-microbe interactions. And then they looked at the impact of a Streptomyces strain that produces a toxic antibiotic compound. So they were looking at what's the influence of that Streptomyces on that microbial community. And while I won't go into the details of that paper, it's very exciting because they were able to show that at different levels, different factors, very complex factors, influenced the phytobiome. So there are many questions that this can lead us to. So why do we care about these things? Well, we care about this because we want to know what factors are influencing the phytobiome content and function, not just the content, but the function of that phytobiome, its ability to be a biocontrol agent, for example, or plant health. And what functions that affect that microbe interactions can be manipulated, for example, to improve soil health or in the health of the plant or to control disease. So many opportunities and many questions that these studies are opening up. We're not just thinking about the phytobiome in the context of plant microbe interactions, but some very exciting studies are emerging now in the insect world. And as we've been promoting this initiative to different societies to get their buy-in and get their voice behind it, what we're finding is that the entomologists got it right away. They see how the interactions with insects and the microbiome are critical to them. So for example, this is a case which was published in PNAS a couple of years ago, where the Colorado beetle actually exploits orally secreted bacteria to suppress plant defenses to the insect so that the Colorado beetle can feed more happily. And if I knew how to activate that video from here, you would see the larvae secreting the nice microbes and feeding on the plant. So we've seen a lot of different papers emerging now with studies on the phytobiomes and the microbiomes since those early papers. And the field is really heating up, and we're learning a lot about how microbes interact with plants and plants interact with microbes. And that allows us to think about what questions we can address. And clearly, the field of questions is wide open. There are ecological questions that can be asked. There are questions about how do the phytobiomes affect plant disease, plant resistance, and plant performance. How do they impact the ability of the plant to take up nutrients? So that impacts those of us who like to eat nutritious plants, and also the animal feed people. They understand that the phytobiome is important for the quality of the food, feed, and fiber that we get. How does the phytobiome impact plant tolerance to abiotic stresses. So if the temperatures go up and down, and does the phytobiome help the plant temper those responses? And from an industrial point of view, what useful organisms or products of those organisms can we mine from the phytobiomes and use in the future? From George's perspective, as the context of this symposia is in soil health, there are many questions that can be addressed from what we heard earlier, that how can the phytobiomes be used to help manage some of those things? And already with the measurements that are being taken, you can understand that there would be different nutritional qualities to the soil. So, for example, could we use the phytobiome or a microbiome to rebuild a depleted soil or to improve a depleted soil if we understood the context or the members of that community? We all hear lots of stories about the human microbiome, and in fact, the Human Microbiome Project has just dominated not only the popular literature, but the scientific literature. And I'm truly amazed by the beautiful artwork that comes with this particular, the human microbiome story. The human microbiome is providing us a nice platform to learn a lot about how we can go forward with understanding the phytobiome of our communities. So what are some of the lessons that we've learned? Well, for example, we've learned that these large-scale consortium-driven projects could be highly effective in learning about microbiomes. And what they've done is they've established not only the infrastructure, but a lot of the technology. Same thing that happened with the Plant Genome Project, which followed the Human Genome Project. The sequencing technologies, the informatic technologies, in this case, the metabolomics, the proteomics, all of that we're going to be able to glean. We don't have to reinvent that. And although the microbiomes exhibit extensive variation across humans, there are some really interesting conserved patterns and profiles that can be recognized. Now, we recognize the complexity that we have more than one host. We have a tree versus an Arabidopsis plant or a soybean, very different hosts in very different environments. We're going to have to adapt in order to glean from that information. The major challenge for the Human Microbiome Project, which we will have to face, is that they're moving from correlative information to understanding causation. And that transition of these longitudinal studies, which are much stronger than the cross-sectional studies, is going to influence how we have to move forward. So the human microbiomes has given us some very interesting paradigm shifts. The commensals are not just passengers. They play critical roles in how our immune systems, our digestive systems, our behavior, and our nervous systems. If we disrupt the homeostasis between the host and the microbiome, we can have, it in and of itself will have a larger effect than the host genetics themselves. And I think that one of the most fascinating things is that the healthy microbiome may not just be a result of the specific set of microbes that are there, but more importantly, the functions that those microbes can carry out. A lot of application, and I'm going to speed up a little bit due to time. You've probably heard about fecal transplantation. This has been enhanced by studies of the human microbiome, and it's established a wonderful foundation for probiotics, the use of probiotics or prebiotics, and the information needed for that. But the phytobiomes, of course, present unique challenges that we haven't seen in the human microbiome. For example, the GI tract is very static in structure, where plants grow continuously. The gut microbiomes exhibit this homeostasis and dysbiosis, but we don't know if the if the phytobiomes attain a homeostasis, or if even this dysbiosis is an appropriate term for the phytobiome. We know that the gut is a host-dominated environment, but the phytobiomes are influenced by other plants, by soils, by animals, insects, or fluctuating environmental conditions. So our level of complexity is much greater than in the human. So this is why taking this systems-wide approach will give us a much, much more robust approach to understanding, for example, how can we manipulate the phytobiome to better control disease or to better make nutritious and safe plants. And there are many opportunities that we have because we're in a plant system, greater opportunities to look at causal relationships, greater power to identify host genotype effects, greater power to associate the phytobiomes and plant health outcomes, and a greater way to enable rapid application of what we learn to the field, because we're dealing with plants and not with people. So to wrap up, our goal is by 2025 to build a foundation to assess climactic impacts. We need the science behind all this to understand interrelationships with nutrient uptake and their utilization, to relate the phytobiome to its impacts on animal and plant health, to safely and sustainably intensify production of food, feed, and fiber, and to literally change the paradigms for plant disease control and crop improvement. So we have a lot of initiatives, and I think you've heard some of them. We're planning a symposium in 2015 that is designed to bring not only these many disciplines, you have to have microbiologists, soil scientists, animal scientists, ecologists, epidemiologists, bring all those people to the table to lay down a pathway or a roadmap for the phytobiomes for the future. And our goal will be to establish this roadmap, this research roadmap. We also are advertising for an APS Public Policy Fellowship for an early to mid-career scientist to work at a high-level government office and to help us promote the phytobiomes, because the Phytobiomes Initiative is about driving funding for research, education, extension towards understanding what's happening in the plant community. I invite you to visit our booth, and we want you to ask your local representatives and your national representatives to add $100 million in new funding to phytobiomes research, so that we can, if you can get these people to work together to do this, we've accomplished a lot in our goal. So contact information here, and I'm going to close with thanking the Public Policy Board members who have helped in all of this effort. And I want to call out Gwyn Beatty, who has been sort of my mentor in phytobiomes and has been very gracious in helping to pull a lot of the thinking together for this presentation and for our grant proposals for meetings. And of course, APS Council and APS Foundation, who are supporting us. Now, here's your job. My job's done. Your job is sitting there. Take out your phones right now. Those of you with smartphones, I know some of you don't have smartphones yet. And tweet your representative. I learned to tweet last night. Tweet your Tweet your representative, and this is an example of how to do it. And if you don't know how, stop by our booth, and my very smart young colleagues will teach you how to do it. Thank you very much.